Hello, and thank you for joining us. I'm Dawn Cullum, the Senior Events and Training Coordinator for Cetera University. As regulators and sponsors, Cetera recognizes the growing importance of model-informed drug development. We are committed to educating the next generation of experts in this field by partnering with eminent scientists at designated academic centers of excellence around the world. This year, our Express webinars are being conducted by our partners and centers of excellence. Today, Hsuan-Ping Chang from the University of Buffalo will be bringing us the final webinar of the series. Ping is currently a fourth-year Ph.D. student in the Pharmaceutical Sciences Department at the University of Buffalo in Dr. Shah's lab. She received her bachelor's degree in pharmacy from National Taiwan University. Then she continued her first MS degree in clinical pharmacy at the National Taiwan University. Her MS thesis focuses on the clinical drug interaction of tacrolimus and everolimus in renal transplant patients. And I'm sorry, I know that I just murdered those names. She worked at the Taiwan Lepson Company as a scientist in the Drug Metabolism and Pharmacokinetics Department. Ping received her MS in the Pharmaceutical Sciences Department at the University of Buffalo in 2019. Her research includes PK-PD of ADCs, as well as the development of physiological-based PK models for antibody drugs in pediatric populations. Today, she will present part of her work about PK and modeling antibody-drug conjugates. Before turning it over to Ping, I have one housekeeping note. We ask that if you have any questions, please put them in the question section. Ping, I'll now turn it over to you.
Okay, thank you. So hello, I'm Hsuan-Ping Chang from University at Buffalo. Welcome to today's webinar, Whole Body PK of MMAE and MMAE-based ADC in mice. So first, let's talk about what is antibody-drug conjugate, ADC. An ADC has three components, antibody, linker, and payload. When you want to design an ADC, each component has its own important consideration. For antibody, it needs to be specifically bind to the antigen that's expressed only in the tumor. For the linker, there are two classes, cleavable or non-cleavable linkers. Cleavable linkers can utilize low pH, protease, or high intracellular glutathione to release the payload. And for non-cleavable linkers, it requires ADC to be internalized into the lysosome and be degraded. For the payload, there are mainly two types. One is DNA targeting payload, such as PBD or SN38. The other is tubulin inhibitors, including DM1, MMAE, and MMF. So the tool ADC we will demonstrate today is trastuzumab conjugate with tubulin inhibitor MMAE using the cleavable protease linker. So another concept I want to introduce for the ADC is drug to antibody ratio, the DAR value. So after antibody is reduced from 1 to 8 of MMAE can attach to the antibody. So the formulation of MMAE-based ADC is often a heterogeneous mixture of antibody with different numbers of payloads. Often we use the average DAR value to represent the overall DAR of ADC in the body. So how does ADC work? So after ADC binds to its target, the ADC antigen complex will be internalized into the cell, and it go to the endosome and go to the lysosome. In the lysosome, ADC will be degraded and release its cytotoxic payload. The payload can exert its cytotoxic effect and kill the cancer cells. So after knowing what ADC is, let's talk about the history of ADC development. So the first antibody conjugate was born in 1957, and the ADC was first tested in human in 1972. In 2000, the first ADC was approved, and now 13 ADC have been approved, with 10 of them being approved since 2017. And actually, nearly 200 ADC have entered clinical trials this year. But why do we especially want to study MMA-based ADC? Because MMA is one of the most commonly used and most studied payloads. 5 of 13 approved ADC use MMA as their cytotoxic payload. Although the success of this approved MMA-based ADC, there are still some challenges in clinics, and we need more study to understand the disposition of these ADC molecules in order to provide a safer treatment for patients. For example, it was found that there are discrepancies in the exposure-response relationship for MMA-based ADC. As shown in this figure, the overall response rate increased with the decreased MMA trough concentration, and the probability of neutropenia increased with decreased MMA trough concentration. In addition, peripheral neuropathy was observed in up to 50% of patients received an MMA ADC, while it was not predicted in preclinical toxicity studies. Furthermore, there is no consensus on which ADC analytes in which biological metrics better represent the toxicity or efficacy of MMA-based ADC. So all of these evidence indicate that only plasma concentration of MMA may not be sufficient to establish a translatable exposure-response relationship, and tissue concentration may be needed to account for. So our goal is to establish better exposure response and exposure toxicity relationships for MMA-based ADC in clinics. To reach our goal, we think the first step is to understand the whole body PK of MMA-based ADC quantitatively. But how? So we will start with PK data and then use the data to build PK models. So today, I will show you how we use the whole body PK data to build a whole body PBPK model for MMA-based ADCs. And as you know, ADC contains large and small molecules. So what PK should we study? The answer is we need to study the antibody PK, the small molecule MMAE PK, and when these two are combined, the ADC's PK. So in today's webinar, I will show you the whole body PK of MMAE and MMA-based ADC in mice. And I will focus on how we can utilize Win-non-Lin to conduct non-compartmental analysis and interpret the whole body PK data. In the end, I will also show you how we use whole body PK data to build a PBPK model for MMAE and MMA-based ADC. So first, let's talk about PK study for free MMAE in mice. Here is the PK study design. A total of 18 tumor-bearing nuc mouse were used, and each mouse will receive 0.1 milligram per kilogram free MMAE single dose. Blood, tissue including heart, liver, lung, spleen, pancreas, kidney, skin, bone, muscle, fat, brain, and also tumor were collected at 5 minutes and also 1, 6, 12, 24, and 168 hours. Tissue and tumor samples were pretreated and homogenized based on the published protocol. And we used the LC-MS to quantify MMA concentration in plasma, tissues, and tumors. And the lower limit of quantification is 25 picogram per ml. So here is the PK profile of free MMA in plasma, tissue, and tumor. So as shown here, the plasma concentration dropped rapidly with only 0.3% of injected dose remaining after 5 minutes. And plasma concentration was below the limit of quantification after 12 hours. the half-life of the MMA is 2.5 hours, which is much shorter than the half-life of MMAE when given as an ADC, which is around 2 days. And MMAE volume of distribution is large, suggesting extensive tissue distribution of the drug. On the other hand, we observe prolonged concentration profiles in tissue and tumor. So the tissue concentration were quantifiable toward 24 hours, except for the liver. The liver concentration is only quantifiable up to 6 hours. And this may be because liver is a primary elimination organ of MMAE. And for the tumor concentration, it decreased only 50% from 1 to 168 hours. So using our observed data of MMAE in plasma and tissue, we can calculate the partition coefficient, the Kp values in each tissue by dividing tissue AUC with plasma AUC. And the table shows the calculated Kp value in each tissue. So the Kp value are more than 1 in most of the tissues, which indicate MMA exposure in tissue is higher than in plasma. And again, suggests that MMA extensively distribute and retain in the tissue. So Kp for red blood cell is 5.5, which is within the literature reported range. And the liver Kp after adjusting for intrinsic clearance is 16. So here we can classify MMA distribution kinetics into 3 groups. First, Kp values are more than 20 in highly perfused tissues, including lung, heart, kidney, spleen, and liver. Second, Kp values are around 2 in poorly perfused tissue, including muscle, bone, skin, fat, and pancreas. Third, brain has limited MMAE distribution, as the drug exposure in the brain is only half of the exposure in plasma. And this may be because MMAE is a P-glycoprotein substrate and can be effluxed out of the brain. So MMAE partition can be explained by blood perfusion rate, cell membrane penetration rate, and the involvement of transporter. And here, I also want to share some non-compartmental analysis experience about AUC calculation and slope estimation. So in Winnoling, AUC can be either calculated by linear or log trapezoidal method. The default method is linear, but this method is more recommended for PD data. And for PK data, the linear method should be used with caution. So as shown in this figure, linear method tends to overestimate AUC for descending PK profile with first order elimination, and the error will be more significant when the sampling interval is large compared to the half-life of the drug. And I will show you the example later using our free MMA data. As shown in this figure, because drug elimination is usually first order, the log method provides a more accurate AUC for PK data, assuming first order decline between the 2 measurements. Of note, log method only applies to the descending curve, and also it is not available when using sparse sampling approach. So for PK data, we generally use a mixture of 2 methods. So as shown in this figure, for increasing concentration, we use linear method, and for decreasing concentrations, we use log method. So here is the table that we compare the calculated AUC for free MMA using either linear or log method. And the full difference of AUC calculated by these 2 methods are shown here. So MMA plasma AUC calculated by the linear method is 2.5 times higher than the log method. And also linear method overestimate AUC in some tissues. In contrast, if we use the model predicted MMA concentration, which has a very dense sampling intervals, the difference between linear and log becomes negligible. And for lambda estimation, although Winnoling provides the best fit estimation, we should always check whether the estimated slope is appropriate. For example, if the slope only includes data in the terminal phase. In addition, when the terminal concentration is quite off, we will use AUC infinity predicted instead of AUC infinity observed. So next, let's talk about the whole body PK of MMAE-based ADC in mice. So before we do the ADC PK study, we produced our true ADC trastuzumab MMAE in our lab. So first, we add T-step to reduce the disulfide bond of antibodies. And then we add MMAE with its VC linker into the solution to allow payload conjugations. We characterize the DAR value by hydrophobic interaction chromatography, and the average DAR for our ADC formulation is around 4. So here is the PK study design for MMAE-based ADC. So mice were injected with HER2 high expressed N87 cells, and after tumor growth, each mouse were injected with 10 mg per kilogram trastuzumab MMAE ADC single dose. Blood, 11 tissues, and tumor samples were collected in 10 minutes, and also 6, 24, 72, and 168 hours. As you can imagine, after ADC injection, there are multiple ADC analytes in the body. And here we quantify 3 ADC analytes. First, we use ELISA to quantify total antibody, which includes antibody with conjugate payloads and NEC antibody. The quantification range is 1 to 500 nanogram per ml. And then we use the LC-MS to quantify unconjugated MMAE and total MMAE. The total MMAE includes conjugated MMAE plus unconjugated MMAE. So here shows the whole body PK profile of MMAE-based ADC for total antibody, total MMAE, and unconjugated MMAE in plasma, tissue, and tumor. Let me show you some key features of the profiles. So first, we want to know whether PK of total antibody of ADC is similar to NEC antibody. So as shown in this figure, we compare total antibody of trastuzumab MMAE with NEC antibody trastuzumab, and the PK profiles are superimposed. This indicates that in mice and in our ADC formulation, the conjugation does not alter antibody PK. And the half-life of total MMAE is shorter than the half-life of total antibody. This indicates after deconjugation, MMAE is eliminated rapidly. And the total MMAE concentration at the first time point is about 4 times higher than the total antibody, corresponding to the major DAR value of our ADC formulation. And the PK profile of total antibody and total MMAE cross each other at 4 days, suggesting DAR value decreased between 1 after 4 days. And the AUC of the unconjugated MMAE is only 0.22% of total MMAE. And the half-life of unconjugated MMAE is around 2 days. This is longer than the half-life when dosed with free MMAE in mouse. So now let's see the tissue PK profiles. So the T maximum of unconjugated MMAE occurs at 6 to 24 hours, and the slope is shallower for free MMAE tissue profile. The slope is shallower than the free MMAE tissue profile as I showed you before. And this indicates formation rate limited of unconjugated MMAE. And in tumors, the 3 ADC analytes show prolonged exposure. So this slide is the highlight of our study. So here we calculate the tissue to plasma AUC ratio, the percent T to P for each ADC analyte. So the percent T to P is calculated by using tissue AUC divided by plasma AUC times 100%. And this table shows the percent T to P for total antibody, total MMAE, conjugated MMAE, and unconjugated MMAE after ADC administration. 2 additional data are also provided. First is percent T to P of free MMAE. Second is percent T to P after administration of NEC antibody obtained from our previous 2 studies. So let's compare the percent T to P between total antibody and NEC antibody. So the ratio between these two analytes is comparable in all tissues. This again suggests that the conjugation did not affect antibody PK in mice. And now let's compare the percent T2P between total MMAE and conjugated MMAE. The percent T2P of conjugated MMAE contribute to more than 80% of total MMAE in all tissue, except in spleen, where the conjugated MMAE only contribute 33% of the total MMA exposure. And this implies that spleen may have higher deconjugation process. And now let's compare the unconjugated MMAE of ADC with free MMAE, which is the most interesting part. So for many tissue, percent T2P of unconjugated MMA correlates with free MMAE. However, there's some significant difference in some tissues. For example, in heart and lung, when giving free MMAE, the percent T2P is high. But when giving MMAE as a conjugate, the percent T2P significantly decrease. In contrast, in liver, when giving free MMAE, percent T2P is low, but when giving MMAE as a conjugate, percent T2P significantly increase. So this suggests that conjugation of the payload with an antibody can alter the tissue distribution profile of the payload. Besides, we also find the percent T2P of unconjugated MMA in spleen are the highest in all tissues, and this may associate with clinically observed adverse event of MMAE-based ADC, such as neutropenia or peripheral neuropathy. And MMA exposure in tumor is 20-fold higher than when administration as ADC. So this confirms that the ADC can specifically deliver the cytotoxic agent to the target. So after evaluating the MMAE and MMAE-based ADC PK data, let's use the data to build PBPK models. So we will first build a free MMA PBPK model and then build an ADC PBPK model. So here shows the PBPK model structure for free MMAE. Figure A is the whole body level structure of the model, and the model includes 16 tissues and a tumor compartment. Figure B is the tissue level structure of the model. So each tissue compartment is further divided into 4 subcompartments, including vascular, endothelial cell, interstitial space, and cellular space. And vascular space is further divided into plasma and blood cell. And the tumor model is shown in figure C. So the MMAE model assumes rapid distribution of MMAE between vascular, endothelial cell, and interstitial space. And MMAE distribution between plasma and blood cell and between interstitial and cellular space is permeability limited. And we use the permeability surface area coefficient, PS value, to describe the process. And the unbound MMAE can partition into the blood cell or into the cellular space. And we use partition coefficient, Kp value, to characterize this process. Plasma protein binding of MMA is reported to be 17 to 29% in mice, and we use 20% in our PBPK model. In addition, MMA is reported to be eliminated primarily via CYP3A4 and 5, and the renal excretion contribute less than 5%. So therefore, we assume MMA is eliminated solely via hepatic clearance. So all the physiological and PK parameters are obtained from literature, and we only estimated PS value in each tissue and the intrinsic clearance when developing the PBPK model. So here shows the MMA PBPK model fitting result. So the figure shows the observed data superimposed with model predicted PK profiles in plasma, tissues, and tumor. The model can capture the data well as the percent prediction error are within 2-4 range. And the PS value are estimated with good precision, and the estimated value are lower than the tissue blood flow rate confirming permeability rate limited distribution. So the model also estimates the intrinsic clearance, which is 137 milliliter per hour. And this is within the twofold difference of intrinsic clearance approximately by using NCA calculated clearance. This free MMA PBK model serves as the first step to build the MMAE-based ADC PBPK model. So here is the ADC PBPK model structure. The ADC PBPK model contains an antibody PBPK model plus a small molecular MMA PBPK model, and they are connected via degradation and deconjugation process. So the upper figure is the whole body level and lower figure is the tissue level structure of the ADC PBPK model. The small molecular MMA PBPK model is adapted from our free MMA PBPK model that I just shown. And this antibody PBPK model is from our previous published platform antibody PBPK model. So the ADC PBPK model mechanistically describes how ADC release its payload. So when antibody is degraded or deconjugated, it will release certain number of payload to the corresponding compartment in MMA PBPK model. And the release number of payload is equivalent to the DAR value at a given time. And since the release of ADC payload is dynamic, we incorporate DAR-dependent deconjugation rate in the PBPK model. And in this model, there are two key assumptions. First, ADC and NEC antibody share the same PK properties. And second, once MMAE is released from the ADC, it behaves like a free MMAE. So one of the advantages of PBPK model is that by inputting all the physiological and PK parameters without estimating any parameter, we can perform a priori simulation. So the figure shows the model predict total antibody, total MMAE, and unconjugated MMAE in plasma, tissues, and tumor superimposed with the observed data. So as you can see, the model can predict the total antibody and total MMAE well, while the prediction for unconjugated MMAE is off in some of the tissues. So we therefore think, what parameters can we modify to improve the model prediction of unconjugated MMAE without compromising the performance of total antibody and total MMAE prediction? So we found that by changing fraction unbound from 0.8 to 0.2, we can improve the model prediction for the unconjugated MMA in plasma and some tissue, as shown in the dashed line. And the rationale for changing the protein binding parameter is that MMA protein binding is species dependent and the reported value range from 20 to 80%. However, more information about payload behavior after deconjugation is required to optimize the ADC PBPK model. So in terms of the application of the ADC PBPK model, we have scaled up our mouse ADC PBPK model to higher species and humans. And in the future, we can utilize this PBPK model to predict clinical PK of MMA-based ADC in different populations, such as pediatrics, or use it to evaluate clinical drug-drug interaction of MMA-based ADC, or to investigate different dosing regimens and optimize dosing strategy of MMA-based ADC for cancer treatment. So in summary, we present the first ever whole body distribution study of free MMA in mice. We demonstrate that non-compartment analysis is a useful tool to investigate whole body PK data of different ADC analytes. And the biodistribution of MMA can be altered when MMA is given as a conjugate. And this also highlights that the importance of investigating PK of free cytotoxic payload in preclinical species, since it is often not possible to conduct payload PK in humans due to their inherent toxicity. And finally, our ADC PBPK model can serve as a platform model for translation and clinical investigation of MMA-based ADC. So I would like to thank my advisor, Dr. Shah, and all the lab members, and the Department of Pharmacy and Pharmaceutical Science, and my committee member, Dr. Balthazar and Dr. Major, the financial support for Center of Protein Therapeutics and NIH grant. So I would love to take any question. Thank you.
And we have a few of them from the audience. The first one is, have you measured the unbound fraction in tissues because distribution of the unbound compound is the one that you should follow?
Let me go back to this. Yeah, thank you for the questions. Yeah, no, we don't measure the unbound fraction in the tissue, and we think this is very important parameter if we want to develop a more accurate PBPK model. But we assume that there may be pseudo equivalence, so we use the partition coefficient, the Kp values, which is calculated by AUC of tissue divided by plasma, and this can represent the unbound fraction between the tissue exposure, because the unbound drug, only the unbound drug can go into the tissue cellular space, right? So we think Kp value may represent this unbound value.
Okay. The next question is, how was dosing of MMAE alone and MMAE-based ADC decided?
Yeah, thank you for the question. So for MMAE, this one is free MMA is 0.1 milligram per kilogram. Actually, we tend to use the highest and those, but these also tolerate those. So I think literature have report the highest dose is around like 0.1 milligram per kilogram. So here we don't really use any model to decide the dose, but we want to use the higher dose that we can. So 0.1 is tolerant in mouse. Yeah, so that's the reason. And for the ADC, because I think also we want to use the higher dose for the PK study, is because if you use the lower dose, you may encounter some TMDD issue. And for a lot of mouse study, they usually use for the antibody use 10 milligram per kilogram, and same as ADC, we decide to use the 10 milligram per kilogram, and this is also easy for us to compare like other studies or different study data. Yeah.
Next question. It is interesting to see the exposure of MMAE changes when given as a free drug versus an ADC. What do you think the reason for that? Okay,
yeah. Thank you for the question. Let me go back to the tables. Yeah, this one, right? Yeah, so this is a very interesting finding for us. So I think for tissue with higher antibody catabolism, it can lead to more payload exposure and result in higher percent T2P when MMA is given as a conjugate. So our data show higher percent T2P in liver. So I think this may be explained by two mechanisms. So first is the presence of the extracellular enzyme, because there are some studies that show extracellular enzyme that is highly expressed in the tumor can contribute to ADC instability and more payload release. And for liver, the second explanation may be the immune cell uptake. So for example, Kupffer cells or other immune cells such as macrophage in the liver can contribute to non-specific cellular uptake of the ADC and increase the payload release. And for the reason why percent T2P decrease when MMAE is given as an ADC, this may be explained by any reduction of MMA-driven tissue uptake. So because ADC can significantly decrease MMA concentration in systemic circulation, the MMA-driven tissue uptake may be altered, right? So this effect may be more permanent in certain tissues such as the heart and lung, as we see in our data. And another significant example is tumor. So via antibody binding and internalization, it can facilitate more payload to go into the cell and bind to the target, the tubulin, and thus increase the percent T2P.
And the last question, what do you think was the difficult part when doing the whole body distribution study of ADC? What are your suggestions for someone who's trying to do that?
Okay. Let me go to the slide, this one. Yeah, thank you for the question. For me, I think for the whole body PK study for ADC, developing a solid analytical method to quantify different ADC analyte is one of the challenges. So for ELISA, you may need to optimize the dilution factor for each tissue, since as you see here, the quantification range is quite narrow. And we also recommend for the standard curve, you should use the same metrics and the same dilution factor as your samples. And for total MMAE, we use pop-pen digestion. So you will need to optimize the condition to make sure you complete digestion and get the total MMAE. And for homogenization, because some tissue is tough and some is soft, so the homogenization method also need to be optimized for each tissue. So for me, the suggestion, I think, at least in the laboratory scale, you should always check your ADC formulation before you're doing any PK study. So you should actually check the DAR profile like this, what we usually do this for your ADC. And after you make the ADC, you should not store the ADC too long before the study. So that's my suggestion.
Great. Well, thank you, Ping, for your presentation, and thank you to the audience for joining us today. We hope that you enjoyed our COE Express webinar series and learned something new along the way. Have a great day.